technique issue. So today I'm going to talk molecular imaging quantification of neurochemicals in cell type specific alcohol metabolism. So when we drink alcohol, most alcohol goes to the liver. That's well known. Metabolized first by the alcohol dehydrogenase, that's the ADH in short. And also the cytochrome P450 2E1, another enzyme, the catalase, oxidize the alcohol to acetaldehyde. And acetaldehyde further oxidized in mitochondria by the aldehyde dehydrogenase type 2, ALDH2 in short. Product is acetate. So in Asia group, we know the, about half of the population has ALDH2 deficient. In those group, when they're drinking alcohol, they have more severe reaction. That's because the accumulation of acetaldehyde in the blood. And but in the brain, we don't know much about alcohol metabolism, and even we don't know which subtype of cells specifically contribute to the alcohol metabolites. And when the human drink alcohol, it has a handover behavior effect, just even the low dose alcohol consumption. But neurochemical mechanisms in the brain underlying this behavior change are poorly understood. Another way is because I doing the imaging study, we know the magnetic resonance imaging of the brain is one of non-invasive imaging technique that can examine, anatomical microstructure and the living function of the brain. I just list here. So MRI has multiple functions and widely used in clinical and preclinical as well. For example, if we use the contrast, we can look in the vascularity in the brain, and also can measure the diffusions and like a function issue, and then the perfusion, the blood flow, if even the tumor diagnosis. And I'm going to talk about the spectroscopy today, used for quantify the alcohol metabolites and the neurochemicals. Here is in patient study. So MRI can see anatomy I just mentioned. So there's alcohol drinking patient that has a severe atrophy of cerebellum, and it's a very prominent damage in the cerebellum. That means the cerebellum is a major brain region targeted by alcohol. And also the function changer, also like a diffusion tension imaging, you can see the patients with alcohol drinking and the control has lost the function connection. And the microscopy also quantified those kind of neurochemicals change in those patients, has showed some important findings in the human study. However, in patients, because it's so complicated, we don't has limitation for the specific gene target studies and the gene regulate the protein's function. So we're going to establish the animal models. So this study is to test whether non-invasive MRS can be used to quantify the dynamic alcohol metabolism, including alcohol metabolites, like I just mentioned, as the aldehyde and also alcohol itself. and the major neurotransmitters such as GABA, which is a key inhibitory neurotransmitter, and the glutamate in mice with conditional deletion of ALDH2 in GFAP positive cell. So here is the model we established in our in the lab as a GFAP ALDH2 flux, and with GFAP crew positive ALDH2 flux, the mice that's specially knock out ALDH2 gene in the brain, especially the astrocytes. That's GFAP for targeted for the astrocyte. And then we also has maybe talk about a little bit in the whole body knockout model, and then the Purkinje cells ALDH knockout model. And in this model, we just give a low dose alcohol, and then we measure the alcohol metabolites and the neurochemicals in the brain. Especially we evaluate the cerebellum in this case by MR spectroscopy, and then we do the behavior test to see the alcohol induce the behavior change related to the cerebellum and the mechanisms. So here is I just mentioned we do those kind of quantification for the alcohol and the acetate and the major neurochemicals, GABA and the glutamate in cerebellum in same animal model. First, we evaluated the ALDH2 messenger RNA expression in different brain region. From here, we know the cerebellum, this one has higher, relatively higher ALDH2 messenger RNA expression compared to hippocampus, thalamus, striatum, and also the prefrontal region, relatively higher. And then the activity showed a similar pattern when we compare with prefrontal as a 1. We can see the significant increase the ALDH2 activity ratio in cerebellum to the prefrontal cortex. And for GFAP image messenger RNA expression also showed similar patterns here. So highest in the cerebellum. That means our model is targeted mainly in the cerebellum astrocyte, and that has a very good correlation between the GFAP messenger RNA and the ALDH2 messenger RNA. So that's for the tissue level, and then we have a co-localization of ALDH2 messenger RNA with GFAP messenger RNA in cerebellum and hippocampus. That's for example to see which cells specifically target or expressed by the ALDH2 messenger RNA expression in which subtype cells. So here we just has two examples for cerebellum and the hippocampus. We do the ALDH2 genes labeled with green. You can see the green colors here, ALDH2, and this is cerebellum. And then the Purkinje cells is here, those red color, you can see. And the blue color, the dapper, is for neuron, and you can see the merged, especially with this ALDH2. It's not much correlate with Purkinje cells, mainly it's GFAP positive cells. That's I think with ALDH2 correlate very well. And also you can see the hippocampus as a similar, relatively intensity expression is relatively lower than the cerebellum, but is correlated well with the GFAP, is not with other kind of cells. So that means the most ALDH2 genes expressed in the astrocytes in cerebellum is highest brain region. And also, though, because we specially targeted in the brain, we examine the liver, because the liver, I just mentioned, is the highest ALDH2 expression, and also so alcohol dehydrogenase also has highest in the liver. So when we do this model, we found that the liver doesn't affected by this transfection. It still has the ALDH2 expression. So this is the wild type, and then this is the transgenic model. So not affect the liver expression, but only in the cerebellum. And then we measure the alcohol injection. We give the different dose. We can see the different dose in single injection at low dose to high dose. And then we measure acetaldehyde level in the serum. We found that there's no difference in wild type and knockout model, and the wild type is no difference. And then in cerebellum also is no difference with the acetaldehyde level in the cerebellum. That's interesting, because most of the people thinking, that's alcohol metabolized in the liver, and then the alcohol metabolized can circulate into the brain. And in this case, I don't see much acetaldehyde accumulated in the brain, in the cerebellum as well. And the hippocampus show the same similar pattern. We don't see the significant difference between the knockout and the wild type. And so this one is just the show the different subtype brain tissue as alcohol acetaldehyde that shows wild type is significantly lower in the knockout model. That means this acetaldehyde cannot further metabolized by this enzyme. That means we have established the ideal model to do this study for the acetaldehyde or acetate affect the human behavior or animal rodent behavior. So there's this ideal model. And then that's
we are running late. Could you please wrap up in 5 minutes?
Okay. And then
thank
we
you.
do the behavior test. We found locomotor is in the knockout model. We can see the significantly increase the rodent activity compared with wild type mouse. And also we do the motor impairment study, and show the alcohol, the injection can cause the motor impairment in transgenic model compared with wild type. And then we inject the acetate, also cause the similar change as well. And to examine to find what cause related to this change after alcohol injection in the cerebellum, we use the MRS. We found that interestingly, low dose and moderate dose injection in the cerebellum, we found the GABA level was decreased compared with wild type, and also the hippocampus also showed a similar change as well, but it's relatively is a higher GABA level. And the acetate, this is the GABA, and the acetate actually in the knockout model, ALDH knockout model, we blocked the acetate level in the cerebellum and the hippocampus. So there's a close correlation, GABA and the acetate. That means alcohol metabolized acetate may contribute to the GABA synthesis, maybe because there has some close correlation, and also GABA correlated with this motor change and behavior change as well. So this also, we measure other kind of neurochemicals. We just mentioned that the GABA and also glutamate also found it's actually an increase in the transgenic model compared with wild type. Tall little bit change, but I don't know the exact function yet. So in summary, we in this study we found LDH2 maybe overexpressed or mainly expressed in the cerebellum compared with other brain region, and we found the alcohol metabolized in the cerebellum contributed to the behavior change, and maybe not from the peripheral alcohol metabolites. And also we found alcohol induced motor function change is mainly mediated by the LDH2 in astrocytes in cerebellum. And also we found that the MRS is a useful tool to quantify the alcohol and the acetate level in the cerebellum in this model. And here we just measured this where's the location, the alcohol location and the GABA location in MRS and the level change. So conclusions, as I already had LDH2 in brain regulate alcohol-induced behavior, and the cerebellum MRS provide directly visualization of quantification analysis of alcohol metabolites and the major neurotransmitters such as glutamate and GABA. And our future study might be use the further study the mechanism of this acetate and the GABA, how cause the behavior change and the motor impairment. So we thank you for our lab researchers and also collaborate with the NIH, Dr. Li Zhang and his colleagues. And also I thank NIH grant, NIK grant, R01 and our university grant as well. Thank you very much for your attention, and I'm very sorry for caused the inconvenience.